Hi, everyone. Welcome to Innovation in Small Molecule webinar series. This webinar series is brought to you by Adesis. I'm your host today. My name is Zhen. I'm the Associate Director for Discovery Chemistry and Business Innovation at Adesis. I have 14 years experience working with a client for the discovery chemistry projects. Adesis is a contract research organization, operates 100% domestically. We provide our clients with services in pharmaceutical, biotech, and specialty chemical industries. Through our innovative science and manufacturing excellence, we respond rapidly to the most demanding organic chemistry challenges. And this helps our client to achieve their research milestones and accelerate their new products. Through this webinar series, we hope to bring you the most recent technology in small molecule synthesis, purification, and analysis. For confidential reason, we're not going to reveal any structure or the client information. And instead, we'll describe the characteristics of these molecules. We would like to invite you to join the conversation with us. We're using a Zoom webinar platform today. And you can use the Q&A feature at the bottom of the Zoom window to type in your question or make a comment during any time of the webinar. And the Q&A feature is where the red arrow is. We will also like to ask you to type the name of the speaker to make the question a bit easier. Towards the end of the webinar, we'll have a Q&A session to answer these questions. As a reminder to everyone, this webinar is being recorded, and we will send you the recording in a follow-up email. Our first webinar series is Innovation in Small Molecule Purification for Pharmaceutical and Specialty Chemical Industry. And this is a three-part webinar series. In the last part, we introduced advanced sublimation technology. If you missed us and would like to know more about this technology, you can download the free white paper from our website. And here's the link to download the sublimation white paper. We'll also put the link in the chat box for your convenience. In the second part of this webinar series, we introduce a new age of chromatography purification, strategy for success. And in the third part, we'll focus on increasing the speed for purification in process and manufacturing. So today, we're very excited to have a joint webinar with Biotouch. The Biotouch works in the field of drug discovery, analytical chemistry, and environmental and water analysis. With customers from pharmaceutical companies, analytical testing groups, and environmental laboratories, Biotouch develops problem-solving solutions to improve workflows while reducing the environmental impact of the process. Our speakers today from Adesis and Biotouch will introduce recent strategy and show you examples of how to maximize the efficiency of chromatography purification. So now I'd like to introduce our speaker today. Our first speaker is Dr. Wei Bao. Wei received his bachelor degree in biotechnology and a master in chemistry from Wuhan University. He came to study at University of Delaware with Professor John Koh, with focus on synthetic organic chemistry and chemical biology. Wei has more than 10 years experience on flash and prep-LC chromatography of small organic and organometallic compounds. Wei joined Adesis after receiving his PhD, and he's currently a senior chemist and a purification specialist at Adesis. Our second speaker today is Bob Bickler. Bob has over 40 years of chromatography experience, with more than 20 years at Biotouch. Bob received both his bachelor and master degree in chemistry from Montclair State University. He worked in application development, technical support, and product design and development at Webman, YMC, and waters prior to joining Biotouch. Bob worked as a product manager for flash purification system and cartridges, and he's currently a senior scientist at Biotouch for flash purification products. So without further ado, please join me to welcome Wei Bao and Bob Bickler. I'll turn the presentation to them.
All right, thank you, Jinjin, for the introduction. This webinar will be presented by me from Adesis and Bob from Biotouch. I will focus on the workflow optimization of a chromatography purification. I will show you some examples with different scale and requirement of chromatography. I will then show some main challenges of chromatography and how to overcome those challenges. I involve the strategies for the plan of the chromatography and the different techniques and instruments. I will have 2 examples to show how to solve real lab chromatography problems. Bob will focus on how to troubleshoot in chromatography and how to translate HPLC data to reverse phase flash chromatography and how to improve the throughput. So before we go into the technical details about chromatography, let's have a look at the organic research and all manufacturers that require chromatography and their common purity requirement. First one is R&D research, why the most time they want a fast deliver or large amount of APIs, mostly in milligram scale with a typical purity requirement about 90 to 95%. Secondly is industrial manufacturer. They usually require a gram to kilogram deliveries, and the most common requirement of purity is around 98 to 99. And besides the overall purity in this area, it's common to have a specific threshold of like some critical impedance. I will present an example shortly. A third example is the area that requires an even higher purity level. In the area of organic semiconductor, usually have a purity requirement about the 99 and very often about 99.9. So let's have a look at Okay, let's just have a look at the 3 specific examples of the 3 specific areas I just mentioned. First is this R&D research example with synthesis and isolate of organic metallic compound in milligram scale. This is a very typical example that happens from academic labs to industry R&D research labs. In this example particular, they synthesize the material containing 3 isomers as well as some other byproducts in the reaction mixture. In order to isolate this 3A, a step of aqueous workup to remove water-soluble reagent and salt, then extraction of the target sample in organic layer, then followed by a normal phase flash chromatography to remove those easily separated impurities. The last step is urine carrier prep LC to separate those 3 isomers. So this general workflow has been used in academic lab and industry for tens of years, and the key factor of the workflow is to use the right tool and method for your purpose. And in our daily life, we always have more complicated problems with each step in this workflow, and you need a specific technique can be used to overcome this problem. I will keep discussing this in today's webinar. So the second example is industrial manufacture of CBD. In this case, the purity requirement is above 99%, and they also have a specific requirement for impurities. The pesticide in this example has to be lower to a ppm level. So the process of this purification begins with the extraction, distillation of this crude material, then subject those material to a flash chromatography and followed by LC purification. So this shown setup on the right here can process tens of kilos of CBD a day with a meter requirement of above 99 and remove of the pesticide. So the last example I want to show is a very specific area of organic manufacture, which is organic semiconductor. In this area, the purity and impurity profile is key to the performance of the final device. As an example shown here, the device performance, which is quantified by the mobility value, is directly related to the purity of the organic compound used in the device. In this article, the level unpurified material is bought from the commercial vendor is already above 99%. But with the further purification, including in chromatography, can increase the purity to above 99.9 to 99.99%. As you can see here, the performance increase with the purity improvement. The example shows here in this article has a 10 to 100-fold increase in performance of this organic semiconductor device by improving the purity. So for all the examples and all our experience in lab, we can see that chromatography is one of the most important steps in organic material making. But there are several challenges it needs consideration for any chromatography projects. First is a purity requirement. The higher purity requirement with the crude material, the more effort it will take. Some critical impurities will need more effort to remove based on their threshold and closeness to the main target material. Second is the speed of the chromatography, including the method development as well. Efficient chromatography will reduce the cost of the label and the time, column usage, solvent usage, waste disposal, as well as there are like environmental impact from making and dispose of the solvent and the columns. Besides the challenging chromatography steps, the analytic step also has a very important impact on this whole purification process. Efficient and accurate analytical method will guide the chromatography to the success. And on the other hand, we have seen examples of chromatography has been redone because of analytical failure to identify the critical impurities. As an example here, a high-res mass or amount will be needed to distinguish these two compounds on the bottom right. So we just talked about the challenges of chromatography, and let's look at the solution we come out to solve those problems. First, we have to look at the whole workflow and figure out in each step if there's anything we can do to help for faster, efficient chromatography. Second step is making sure we have accurate, efficient, and very easily accessible analytical method for checking the purity. And the analytical method also has to be reliable. So the last step, which is the one we will focus on today, is a method development for chromatography with the right tool. So let's look at the whole workflow first. This is a very typical workflow most chemists use, from synthesis to workup to purification. So after workup is method development for flash column or prep LC. The choice between flash column and prep LC is very project specific. Prep LC has a high theoretical plane number, better resolution, but a lower loading capacity. The purity requirement usually dictate which one to use. There's some general specs of a common bench top prep LC and a flash column for you to as a guideline here. So as a first step of the workflow, workup as a pre-chromatography step can help to refine the product and remove the interfering solvent. Next step after the workup is to find the best method for chromatography. Method development for flash chromatography usually start with for flash chromatography usually start with a TLC or TLC mass spec. For prep LC master scouting, HPLC with analytical column with the same packing material as a prep LC column is usually used. Bob later will also show you a product they can translate HPLC data to reverse phase flash column chromatography with high accuracy. Sometimes on the bottom of this workflow, you can see we pre-run a flash column for the crude materials and transfer it to prep LC. This also can help to narrow down the impurities and focus on the close separations in prep LC. So as a part of the workflow optimization, workup optimization can increase the efficiency of the whole process. The workup typically includes steps like in extraction, filtration, and use a metal scavenger, and distillation, trituration, recrystallization technique. For some small to medium scale workup, some commercial product can work up your sample in times of seconds compared to hours using a traditional method. Like this commercial workup cartridge, have a picture here for extraction and metal scavenger. This is particularly helpful if you have multiple samples to process in one time. As a more advanced tool compared to TLC for flash column method development, TLC mass spec develops flash column method much faster and more accurate. Chemists can identify the peaks mass on the TLC plates in a couple of minutes compared to the traditional way where they have to elute all the peaks in the flash column and check the LC mass for all the peaks and go back to and then roll drive after hours of waiting for the LC mass data. So here is a video demonstration of how TLC mass spec works. So as you can see, the chemist finished one TLC, and he put this TLC in this machine, and the machine have a nozzle can check any spot on your TLC plate, then inject to a mass spec, direct injection. And in matters of like 1 or 2 minutes, you can get the mass information data back. And after the mass back, you can see here, it takes about 1 minute. Then you can continue to next spot if you want after cleaning of the nozzle here. So after you have all the information for, in this example, 3 peaks, you can further optimize your flash column method to accelerate your chromatography. So now let's talk about the flash column method development after the TLC mass spec. So there are 3 aspects that chemists can use for scouting: the stationary phase, the mobile phase, and the detection and collection method. Choice of a stationary phase depends on compound properties like polarity, function groups like amine or acid, and hydrophobicity difference if you want to use the reverse phase. The mobile phase then will be decided after the choice of the stationary phase. Not only we need to choose the proper solvent strength so the target product compound have a proper retention time on the column. We also want to consider the solvent selectivity. Here's a triangle of the solvent selectivity groups that can help to choose based on their selectivity. Bob will later explain this one in detail. So the detection and the collection is another one method usually less discussed in chromatography, but also very important. UV is the most common way. ELSD is a supplementary method mostly used when there's no chromatography with the compound. Mass-directed collection and detection can be really helpful in a lot of cases. There's a choice you can choose a mass-directed collection based on the target and impurity ion ratio with the mass spec, which can help you to collect the product in a higher purity portion. So I will turn into Mac to Bob, and he will explain more about the method development of a flash chromatography.
Thank you, Wei. So Wade talked about a little bit about flash chromatography and method development. So flash chromatography is kind of the first step within the workflow for chemists to purify what they've synthesized to remove impurities that were generated during the synthetic process. And so Wei mentioned stationary phase selection as being critical here, and it's one part of several that go into optimization of a good flash chromatography method. And this doesn't take a lot of time. TLC is very helpful here, but it's important to understand the interactions between the compounds in the reaction mixture and the stationary phase. So typically, if the compounds tend to be on the polar side, then they're going to be attracted to polar media like silica gel. And that's typically known as normal phase chromatography. Likewise, if you have lipophilic compounds, they're going to be better attracted to hydrophobic type stationary phase, and that's typically reversed phase. Organic amines can be problematic. They're strongly attracted to silica gel because the amines tend to be basic, whereas the silica surface can be acidic. It's a Bronsted acid. And so it's usually better to try to purify these types of molecules using a similar type of stationary phase, an amine functionalized or bonded silica gel. So along with stationary phase selection just based on the chemistry of the molecule, another aspect is the solubility of the crude mixture in a particular solvent. So if you've done your reaction, your compounds remain soluble in the solvent in which the reaction was conducted, that'll help you with determining which way to go. So if you've done a reaction at high temperature, let's say using DMSO or DMF, you're going to maintain solubility. Reverse phase, not a bad way to go. Typically, it requires less workup. Likewise, if you've done a synthesis, say in toluene or methyl acetate, something like that, then normal phase will likely be the more appropriate methodology. And so besides looking at stationary phase and compound solubility, in the compounds chemistry, chemists will also, depending on solubility and the methodology, consider how they're going to introduce their sample into the column. And there's two typical ways to do this. One is a liquid load, where you take the solution containing your reaction mixture and you load it directly with a syringe on top of the column, or in situations where the best solvent is too strong for the mobile phase in the method, dry loading becomes a viable option, in which case you dry your reaction material on a sorbent and remove any solvent from it. Along with that, then there's the decision whether to run an isocratic purification, which from start to end consists of the exactly same solvent ratio, or running a gradient where over time you increase the solvent strength to help elute more retained compounds. And to get to that decision, you really need to do some preliminary scouting work. As Wei had mentioned previously, primarily using TLC, especially if you're going to go down normal phase purification strategies. So you can use TLC. With normal phase chromatography, you have many solvents at your disposal. And most chemists will typically use 4 solvents, hexane, ethyl acetate, or dichloromethane and methanol. But as you can see on the table here, there's a broader list than that. So Wei showed that triangle of solvent selectivities. And one corner is a proton acceptor, the other one is a proton donor, and the third is a dipole. So what I've done here is eliminated those 3 aspects, but have consolidated solvents into various selectivity groups. So all your alcohols are in the same group, acetone, acetonitrile alpha acetate are all in the same group, et cetera. So to do this method development, chemists can really do themselves a valuable service by running parallel TLC using a variety of miscible solvents from different selectivity groups, because just because you can't get a separation with one set of solvents doesn't mean you can't get that separation accomplished with a different set of solvents. And so once you've got that selectivity determined by the proper selection of solvents. Then, as Wei mentioned, then we have to look at solvent strength as well. And you can see in the table here, they rank from a low of 0.01 with hexane all the way up to methanol at 0.7. And this is an important aspect of the method development process. The reason is that just because you get a separation on TLC doesn't mean it's going to translate to flash chromatography. So if you have a separation of your compound of interest from an impurity or two, but it elutes fast or high, gives you a high RF value or a retardation factor in TLC, then if you have high RF for your compound and the byproducts, then you're going to get fast elution from your flash column. And so it's important to control solvent strength in order to maximize compound retention and facilitate a better separation. So typically we're looking for with TLC to get the compound of interest, your target synthesized molecule, within a range of 0.15 to 0.4. And as you can see on the far left, we have an example where we have two compounds well separated, but with high RF values, and the problem is they elute too early. By weakening the solvent system to get compound B within that 0.15 to 0.4 range, we've now increased retention, which means there's more interaction with the stationary phase, less solubility in the mobile phase, so we can provide or generate a better separation, which means that we're going to get higher loads and higher purity fractions. And in the example on the far right, we've got both the product and its leading byproduct in that zone, and which is going to maximize compound retention and also maximize purity of the resulting fractions. So once you've gone ahead and selected your mobile phase from a selectivity perspective and a solvent strength perspective, there's another choice to make. And so that's in the silica gel properties itself. So traditionally, flash chromatography, at least normal phase, has been performed using a granular silica gel with roughly 40 to 63 micron particle size, 60 angstrom pore, and 500 square meter per gram surface area. And so for most separations, that's adequate. However, as you can see here, we don't get as good a separation as we would like with that particular material. And so what Biotage has done over the last few years is introduce other silica gels with higher surface area. And these media with higher surface area provide increased floating capacities and increased retention. And as you can see here, just by moving from a granular 40 to 63 micron, 500 meters squared per gram silica gel to a similar particle size media with about 45% higher surface area, you can see that retention has increased and the separation has improved. Now, if that's not good enough, then the chemist always has the option of moving to a 20 micron version of that same high surface area silica gel, which then improves the separation even further, to the point where, depending on how good that separation is, the chemist may be able to actually reduce the column size and achieve the same purification level. And in doing so, then would reduce the amount of solvent consumed and that needs to be disposed of. So amines, I mentioned this a little bit earlier, they can be highly troublesome to purify using silica gel. A lot of chemists will use silica gel, but they resort to dichloromethane, which is a toxic hazardous solvent, with methanol, and then oftentimes add a competing amine like ammonium hydroxide. And even with that, they can do their TLC, translate that method to flash, and not get the results that's desired. Working with this sort of a solvent system can be very tricky, and you don't always get the results that you want. So a better approach, as I mentioned previously, is to have a stationary phase with similar chemistry to the molecule you're trying to purify. And so using an amine functionalized silica gel, we now have a surface that's more tolerable of organic amines and typically uses solvents such as hexane and ethyl acetate, or maybe ethyl acetate and isopropanol. And as you can see here with this mixture of tricyclic amines, all that have got high pKa's, not separable and have very long retention on silica gel, but easily separated on the amino material. And so we've covered so far method development in a normal phase perspective, using thin layer chromatography and then translating thin layer chromatography to flash chromatography. But there's another aspect that we need to talk about, and Wei will pick up on that, which is reverse phase chromatography. Wei?
All right. Thanks, Bob. So in this second part, the reverse phase, I'll start with reverse phase chromatography with PrepLC. So PrepLC method scouting shows the same theory as a flash column method development. So with a higher theoretical prime number of a PrepLC column, PrepLC is mostly used in situations that with different difficult separation of impurities that a flash column sometimes have problem with. Analytical or semi-prep or scale HPLC with same packing material as a PrepLC column is mostly used for method scouting. So similar to flash column, the variables for PrepLC method scouting is like a stationary phase, mobile phase, separation method, and also detection. So in the stationary phase, you have the choice of the C4, C8, and amino-cyanophenol. Some can be used reverse, some can be used for normal phase, some can be used for both. And the mobile phase, and also the separation method, which usually normal phase or reverse phase, and sometimes it is helical. And for extension to the normal phase, we have SFC and a chiral separation. So the choice of the detection method usually is UV, LSC, and mass direct, as I talked before. So we can directly transfer any HPLC analytical data to PrepLC, and but with larger scale PrepLC purification project and method scouting for PrepLC is well worth the time. With a tailored method for PrepLC, we can save time and cost for the purification. So traditional HPLC and the UHPLC can be used for PrepLC method development, but usually in those systems, solvent solubility, solvent scouting ability is very limited due to the number of solvent it can be used in the same time in the same machine. So here's a picture of a specifically designed HPLC scouting system with more solvent for each pump and ability to have a different composition of the solvent, coupled with HPLC highly sensitive detector and a multiple column choice. We can scout like hundreds of solvent column conditions in just one setup. So here is a software diagram of this example HPLC scouting system. So each pump have 4 solvent lines, ability to deliver a combination of the 4 solvent in any percentage. So in this particular setup with additional switch box for pump 1, you can have 11 types of solvent in total and about 270 solvent combinations for you to choose in this setup. We can let the instrument run like 24/7 if you need it. Afterwards, you can directly transfer the scouting method to PrepLC with PrepLC's column. So I just briefly talked about the method development for PrepLC. So Bob will talk about using HPLC for reverse phase flash column chromatography.
reverse phase chromatography is used at times because normal phase can't do everything. And so Wade just talked about utilizing an HPLC to do some method scouting and method development work for transfer to preparative scale chromatography. Well, flash chromatography is a form of preparative chromatography as well. And there's hundreds of brands of columns on the marketplace, most of them with small particles and different types of underlying silica gel and different chemical and physical properties. And so while a chemist could, in theory, use a generic HPLC column, do some method scouting and transfer it to flash, chances are, just like Wei talked about when running, moving from HPLC to preparative HPLC, you want to match stationary phases and media. You want to do the same thing with flash chromatography. So best approach, if you're going to use an HPLC to do your method development, is to get what Biotage calls a scaling column, which is an HPLC column that's filled with the same exact media that we use in the flash column. This way the selectivity is identical and the column efficiencies are identical as well. And there's, I'll go into more detail with that a little bit later. Okay. So in developing a method for reversed phase, you still need to do some scouting, as Wei mentioned, with the automated HPLC system. And the reason we want to do this is because not all solvent blends in reversed phase will provide similar separations or provide you a separation of every typical type of sample. And so it's good to be able, just like with normal phase, to evaluate different solvents in different ratios. As we see on the left here, a couple of examples. Is on the left, we have a situation where, well, water and methanol fail to provide adequate separation between the compounds. That water acetonitrile gradient had success. And on the right-hand side, we see just the opposite, where the acetonitrile water gradient did not provide a lot of selectivity and separation, and the water methanol gradient had some success in doing so. So it's important to evaluate different solvents in different ratios with reverse phase as well as normal phase. For cases where your compounds are charged and ionizable, that presents a different set of issues, and chemists will typically know that because they're manufacturing the particular molecule. So if you have this type of situation, if you run the chromatography in an unbuffered environment, chances are you're not going to get acceptable chromatography, as we see here with these food colors. Without buffering, compounds dilute early, they're broad, they tail, and you're getting compound 1 going into compound 2 blending into compound 3, etc. So need to address the ionization by adding either an acid, a base, or a buffer to tie up those ionized sites, make the compound more lipophilic. If it's more lipophilic, it'll stick to the stationary phase longer. And because it's not ionized anymore, the equilibrium is shifted to the non-ionized state, peak shape improves. So we actually get better retention, better selectivity, better purity fractions. And so early on, I talked about using the scaling column to develop a method and then transfer that to flash chromatography. And I have an example right here for you. So we have a mixture of 7 different compounds here, and on an HPLC, I use the scaling column to develop a particular method. So HPLC is done in a time format. Flash chromatography is performed using a term called column volume. So column volume is the amount of empty space in a packed column. So if you pump solvent through the column, it is the amount of solvent that occupies the packed bed. And so in an HPLC column, you can determine that quite easily by multiplying the void time that accompanies the test chromatogram with the column by the flow rate. And so in this particular column with this 4.6 by 250 millimeter dimensions, the void time at 1 milliliter per minute was 2.64 minutes. So that means its column volume is 2.64 milliliters. And so when it comes to developing methods, you can think about developing your HPLC method in terms of column volumes. And typically you're looking at starting off with one column volume under a certain set of conditions, weaker conditions, and then running a gradient from that to a higher concentration or higher strength solvent ratio over about 10 column volumes, and then hold that for 2. So with that in mind, when I built the method, I did 40% methanol for 1 column volume, or 2.64 minutes, 40 to 100 over 10 column volumes, or 26.4 minutes, and then 100% methanol for 2 column volumes for 5.28 minutes. And you can see the chromatography there using the scaling column provides pretty much a full separation of all 7 peaks. So just simply transferring that over to Flash and utilizing exactly the same method, we get the same chromatography. Now, differences are, we have a column volume on the HPLC of 2.64. On the Flash column, it's 17 milliliters. What I haven't talked about is linear velocity. So linear velocity can play a key role in how good a separation is, because it will determine or impact mass transfer kinetics. And so linear velocity can be determined by dividing the column length in centimeters by the void time. And so with the HPLC column, we're at 9.5 centimeters per minute. And it just so happens that 30 milliliters per minute on the Flash column, we get the same linear velocity by and large. So the mass transfer kinetics hold up, and we end up getting the same selectivity, the same separation, but with a much higher sample load. So we're going to show you some examples of how this all works together. And we have 3 case studies, and I'll tackle the first one here. So one of the aspects of chromatography and being a good steward to the environment is minimizing the amount of solvent that we consume and therefore dispose. And so part of the method development process will help in that regard. And so in this particular reaction I ran in my lab, the amide produced is relatively polar. So hexane ethyl acetate on silica was unsuccessful. So I had to resort to some other solvent systems. So as I mentioned earlier, dichloromethane with methanol, very popular solvent mixture for polar organic molecules. Problem is that the method you create on TLC doesn't always translate to results with Flash. And you can see that we get no separation of the product and the byproduct using DCM methanol. Using DCM and acetonitrile, remember, acetonitrile, totally different selectivity group than methanol, we actually get a partial separation in the byproduct elutes prior to the product, but it's not fully resolved. A much more environmentally friendly solvent blend for to replace DCM and methanol, or even DCM and acetonitrile, is using ethyl acetate and IPA, typically in a 3-to-1 mixture with hexane. And then you can see here, we got a great separation between our product and the trailing byproduct. And so for a normal phase separation, this is by far the best of the three, and we're not using as nearly hazardous solvents as with the TCM, methanol, or DCM acetonitrile blends. But taking this a step further, just another situation where reversed phase comes in handy, in that we are able to do the purification using much more environmentally friendly solvents, water and alcohol. And not only do we get a full separation of the product from the byproduct, we actually can recover some of the starting material as well. I'll now pass it over to Wei to talk about case study number 2.
Okay, thanks, Bob. So in case study number 2, we're going to look at the PrEP LC, MS scouting, and how it helps in lab. So this is an example of a purification organic metallic compound. It has very low solubility in water and a high purity requirement, so about 99.9, and the solubility is very limited in this analytical method. So we need 200 injections for 8 grams of crude with original purification method. So the original purification method is directly transferred from this analytical method using C8 and water acetonitrile with 20 ml per minute. So instead of doing those 200 injections, we did a method scouting to remove this critical impurity here. So after some scouting, we developed a method with using a C18 instead of a C8 with 80 milligram per injection because of this solvent change from methanol, from water acetonitrile to methanol acetonitrile. So it's non-increased reverse phase purification. So we were able to shrink the runtime from about 25 minutes to 3.5 minutes when they can still retain the same separation. So the result is we separate the compound to high purity, about 99.9%, and save the runtime by 14-fold from 18.4 hours from one machine runtime to 6 hours, and save the solvent usage by 7-fold, and reduce solvent waste from 100 liter to 14 liter, and save about $4,000 alone in this project for solvent purchasing. But most importantly, we reduce the environmental impact and the cost to dispose of this waste. We cut the emission of CO2 from 167 kilograms to 25 kilograms, and save around extra $4,000 on solvent waste disposal. So in case study 3, this is more comprehensive project. So this is a partnership with a biotech company developing breast cancer therapies using small molecule. So in this particular project, we have some challenges, including some intermediates are unstable during aqueous walkup. So there's a high polarity final targets and intermediates. So normal phase might be challenging. And the final targets are decomposed with acid or neutral modifying. And there are some undesired diastereomers inseparable on LC mass. So the solution for all these challenges, we use this carbon resin, this ion exchange resin to avoid the increased workup. We did some PrEP LC and a Caro study, and we separated some compound using PrEP LC. And for some other compound, we using this amino D, basically it's amine containing silica for method development and the separation. And for this diastereomers, we using Caro HPLC and confirm the result by OMR. So the results of all this solution, we increase the average recovery from 3% to 50% from synthesis. So that reduced a lot of material needed for synthesis, and reduced around 80% the cost of raw material, and reduced 80% of the waste, and minimize the undesired diastereomers. So in total, we were able to deliver more than 100 compounds, which all need 10 to 15 synthetic steps in 8 months with desired purity, and help the client achieve the milestone. So of all these techniques I just talked, the only downside is the cost of the setup. So the initial investment of this could be very high, and they also need a dedicated space with no interruptions. And you also need a chemist with proper training and experience. And actually that's the most important factor for the final outcome. So in today's webinar, we have a look at the whole workflow of chromatography. We talk about a flash chromatography and the normal phase method development, reverse phase method development, trap LC method development. And I will turn mic to Jinjin.
Well, thank you, Wei, and thank you, Bob, for a very nice seminar. So now we are at the Q&A session now. As a reminder to our audience, please use the Q&A feature at the bottom of the Zoom window to type in your question instead of using the chat box. And also, please tag the name of the speaker when you ask the question. We have our first question actually coming from the chat box. So the question is, how long does it normally take for you to develop a prep LC method with HPLC method scouting? So I think this question is for Wei.
Thank you, Ginger. I think this is a very typical question and a very good question to ask about this prep LC method development. So a prep LC development with HPLC, usually once you have analytical data and have a good understanding of the solubility, like 2 or 3 tries of with HPLC or the scouting system, you were able to get a really good method. For some really challenging and with a high purity requirement, you can let the machine, you can set up like maybe 10, 20, maybe 100 methods at one time. That maybe take roughly maybe overnight to 1 day to finish. So the question to the answer, I would say like from a couple hours to maybe 1 day to get a really good result for the method.
Thank you, Wei. Our second question is, why should I consider reverse phase chromatography? So Bob, would you like to address this question?
I'd be happy to. So reverse phase chromatography is typically utilized in situations where either nothing works, at least from a normal phase perspective, in separating the target molecule from some impurities that, regardless of the solvents that you use, just won't separate. And so reverse phase oftentimes will solve that particular problem. The other reason to go with reversed phase initially would be if your reaction mix is soluble in polar solvents, methanol, water, DMF, DMSO, NMP. Those solvents are not compatible typically with normal phase chromatography. They're just too strong and they'll displace the compounds from the sorbent when you inject your sample into those systems. So they're best suited to reverse phase chromatography. Hopefully that has answered your question.
Thank you, Bob. We have another question for you. So what other substrate can be conveniently purified over a NH2 modified silicon normal phase?
So not sure I understand that question. So normal phase, if I understand it correctly, what sort of compounds are best purified using normal phase chromatography? And anything that is got a little bit of polar functionality, typically neutral compounds or slightly acidic, that are soluble in organic solvents, DCM, toluene, ethyl acetate, acetone, more times than not lend themselves to traditional normal phase chromatography using silica gel. The amino materials, immunostationary phases are designed for organic soluble secondary, tertiary amines and nitrogen heterocycles. Hopefully that has answered the question.
All right. Thank you, Bob. Well, thank you, Wei, as well. So with that, we are at the end of our webinar. So I want to also thank Logan, Sarah, and Rick from Biotouch for this webinar. So, well, thank you, Logan. You just show up. I also want to thank all the audience for joining us today. If you have any further questions about chromatography purification, you can reach out to us through this email address. We also put the email address in the chat box. So in the next part of this webinar series, we'll talk about increasing the speed for purification in process and manufacturing. So please stay tuned for the next webinar. You can also check our website at adesisinc.com or follow us on Facebook and LinkedIn for any future webinars. So thanks again for joining us. We hope to see you again soon.